This video review will cover amino acids and protein purification.
Let's get started with amino acids. In the main chain of amino acids, we have our amino group, our carboxylic group, and our R group, which determines the stereochemistry. Amino acids naturally occur in the L configuration. Looking at the main chain of an amino acid, we have our amino group, which has a pK of 10, and our carboxylic acid group that has a pK of 2. Depending on the pH of the solution, the amino acid or the carboxylic acid can be protonated or deprotonated. Now let's go through the different R groups. You will need to be familiar with the name, 3-letter abbreviation, and 1-letter code, as well as any side chain pKa's mentioned. First up, we have our non-polar side chains. They consist of tryptophan, phenylalanine, lysine, alanine, valine, isoleucine, leucine, methionine, and proline. Proline creates a unique structure as it binds to the backbone of the amino acid, creating a ring. Then we have our polar side chains. They consist of tyrosine, which has a pKa of 10, serine, threonine, asparagine, glutamine, and cysteine that has a pKa of 8. Next, we have our basic side chains. They consist of lysine that has a pKa of 10.5, arginine that has a pKa of 12, and histidine that has a pKa of 6.04. Lastly, we have our acidic side chains. We have aspartic acid with a pKa of 3.9 and glutamic acid with a pKa of 4.1. For a little review, list the following amino acids in order of highest to lowest pKa at a pH of 7. Pause this video to attend to this question. The answer to the question is lysine, tyrosine, cysteine, and glutamic acid.
For the second portion of this video, we are going to look at the purification of proteins. We will examine how to obtain proteins from cells, and then we will discuss different chromatography techniques to purify proteins. To obtain proteins from cells, we must create a crude extract by lysing the cell and then subjecting them to differential centrifugation to separate all the components of the cell. Once the supernatant with soluble protein is obtained, a salting out procedure, typically with ammonium sulfate, is used to precipitate proteins from the solution. Once we have isolated our proteins from the cell, we need to purify the proteins, likely to obtain a protein of interest. Chromatography techniques are the most common and useful way of separating and purifying proteins. Column chromatography takes advantage of differences in protein charge, size, binding affinity, and other properties to facilitate separation. Based on the construction of the column's stationary phase and mobile phase used, some proteins will migrate faster or slower depending on their interactions with the column construction. In ion exchange chromatography, proteins are separated by their differences in the sign and magnitude of the net electric charge at a given pH. The column matrix is a synthetic polymer containing charged-bound resin. A column with an anionic resin will retain cations, and the reverse situation will retain anions. A buffered mobile phase carries the ions through the column, then a salting buffer will elute the retained ions. In size exclusion chromatography, we separate proteins according to their size. The stationary phase consists of different sized polymer beads. In this method, large proteins elute from the column faster than smaller ones, as small proteins get retained by pores within the stationary phase. The stationary phase in affinity chromatography are beads with covalently attached ligands that bind to a specific protein. As a protein mixture is passed through the column with a buffered mobile phase, the protein of interest binds and is retained within the column. Another way to purify proteins, although uncommon, is through electrophoresis. In gel electrophoresis, proteins are separated based on the movement of their charge in an electric field. To give an example problem of this section, say that we are given a mixture of 3 proteins, A, B, and C, with the details shown. Pause this video and describe a method that could isolate each protein. One method to accomplish this isolation would be to conduct size exclusion chromatography. Doing so would isolate protein B with a molecular weight of 60 kilodaltons and leave a mixture of protein A and C, since they both have a molecular weight of 45 kilodaltons. Then you could treat the mixture of protein A and C to ion exchange chromatography. Based on the differences in net charge, each protein will then be isolated.
Thank you for watching this review of amino acids and protein purification.
For more information, read Chapter 3 of our Principles of Biochemistry textbook.